Hello, everyone, and thank you for joining us today for another ACE Spotlight presentation. My name is Rouzbeh Afrasiabi, and I will be your host. Today, we have Mario Krenn, who is a postdoctoral fellow at University of Toronto, Vector Institute, Department of Chemistry, and also Department of Computer Science. Today, he will be talking about his recent work titled SELFIES, 100% Robust Molecular String Representation. With that, and without further ado, I hand things over to Mario. Mario, please take it away.
Thank you very much. Thanks a lot for the invitation. Yeah, I will talk about a new representation for semantically constrained graphs that we developed recently called SELFIES. And this work I did together with my colleagues Florian Hesse, Akshat Nigam, Pascal Friedrich, and Alan Aspuro-Gusic at U of T and Vector Institute. So the bigger picture of the question that we want to investigate can be summarized by two questions. So first, how can we make a machine learning algorithm to outperform human scientists in some interesting tasks? And second, if we did this, how can we learn what the machine has learned? So what did the machine learn that the human didn't yet understand? And in my talk, I will cover mainly the first question and then show one rough idea about the second question, which I find extremely interesting, learning scientific insights from the machine. Okay. So the first question is roughly the answer will be machine learning by design of functional molecules. So that is what I will focusing on. And roughly one can think about it. Some examples are solar cells, batteries, or drug candidates. Okay, so SELFIES is a different way of representing graphs. And so first, what is a graph? A graph is a simple collection of vertices that are connected by edges. So this is a very broad concept, and a large amount of structured data can be represented in a graph as graphs. And one example for large graphs are also called networks, are so-called social networks. Here, every vertex could stand for one person, and every edge could stand for their relation, for instance, whether they are friends or not. In the natural sciences, graphs also play a huge role. But there, often you have data that is additionally constrained. And those constraints come from the specific domain that you are in. And I show you some examples. In biology, for instance, you can think about RNA or DNA also as a type of graph. In this case, tRNA, where each nucleobase could be now your vertex, and each bond between nucleobases could be your edges. In this case, we have a constraint that comes directly from the chemistry and from the physics, namely that the nucleobase A can only bond with U, and G can only form bonds with C. So this is a semantic constraint that that appears in this domain. In a different field, in chemistry, we can represent molecules as graphs, so-called molecular graphs, and we also have additional constraints on those graphs that comes from the scientific domain. Namely, in that case, every vertex stands for atom, and the edges stand for connections between bonds between the atoms, and the number of bonds that one atom can form depends on the atom itself. So, for instance, an oxygen can only form 2 bonds. A hydrogen can only form 1 bond. Carbon can form 4 bonds. And this constraint directly comes from physics. So we have graphs that have additional constraints in that case. A third example could be physics, where abstract graphs where each vertex could stand for an optical element. And in that case also we have constraints that comes from the properties of the elements. For instance, a beam splitter can have 2 input modes and 2 output modes. That means maximally 4 connections, while a detector has only 1 input. So there you also have additional constraints. And those constraints, those semantic constraints, is what we will deal with our representation. Now in the talk, I will mainly focus on chemistry. Okay, so the first question is how chemical graphs usually represented on a computer. So that question goes back to the late 80s when David Weininger introduced a very simple but still until today de facto standard representation of molecules. And that representation is called SMILES, and the idea is quite simple and clever. So the idea is that you have a main string that goes through the molecule, which you can represent as a sequence of atoms. And here, this string has, this main string starts with a nitrogen. There's an implicit carbon, another implicit carbon, another nitrogen. That is represented here, nitrogen, carbon, carbon, nitrogen, and so on. So that is the main string, and what exists in addition are branches. So and here a branch from this main string can be represented by brackets. So here this is a branch. It has an implicit carbon, another implicit carbon, which is written in bracket as carbon, carbon. And the last remaining piece are additional bonds that can form rings. For instance, here we have a ring, and this additional bond is shown as a number 1 from this bond to 1 to this atom. So this payoff number represents 1 additional bond. And with that, a huge amount of chemical space can be represented. Okay, now we know how to represent molecules in the computer. We can now try some generative models to design molecules. So a problem is that our molecular representation is discrete. That means we cannot use standard continuous optimization techniques that we know and use in machine learning. So we need to do something different. And 2 years ago, there was a very beautiful idea that solved that problem. The idea is that you take your SMILES representation and you encode it into a continuous space, and you decode it back to a discrete representation. And this encoder and decoder, they together form a variational autoencoder. And and as an effect, we have our representation of the molecule transformed into a continuous representation. So we start with a discrete representation, we go into this continuous space, and we can go back into the discrete representation. And now, when you have your continuous representation, you can add a third neural network that gets as input a point from this continuous latent space and has to predict properties of the molecule. So in the training procedure, it works like that. The variational autoencoder itself is trained by giving example and giving many examples of molecules, and it should encode and correctly decode, reconstruct that molecule. So the encoder and decoder need to find a way to represent this discrete structure into this continuous space. And in addition, this third neural network is trained by giving examples of properties of these molecules. That could be, for instance, how solvable is the molecule. And now this network learns from a given point in this continuous representation to predict the property. Now, after the training, after the reconstruction works well, and the property predictor works with a sufficiently high quality, one can actually invert this network and try to find a region in this property space that has a very high property. So what we do is we scan them in this continuous space to maximize the output of this prediction network. And when we do this, after we finish the optimization and we find some point in the latent space that seems to have very high properties, we can use now the decoder and decode this point and see what is actually that molecule that is represented by this continuous point. And in that way, we are creating new molecules, because potentially the network has never seen this specific point. Okay, so with that, we can find new molecules and maximize their properties. Now, I told you before that our representation could be smiles, which has many advantages, and for that reason, it's used for the last 30 years. Like, it is human readable, it can represent enormous chemical space, it is quite simple. But it has a huge drawback, namely, it is not robust. That means a large fraction of strings are actually invalid. They don't correspond to valid chemical graphs, or even to valid graphs at all. This can be seen in a very simple example. There are two different types of mistakes that can happen. The first mistake is that a string could have something like a ring starting element, but no ring closure element. So that string is not valid. It doesn't correspond to any valid graph. A second example could be that a branch is apparently opened but never closed. So that string also cannot be represented or interpreted as a valid graph. And now those syntactic mistakes, there's a additional mistake that can happen, namely semantic mistakes. Semantic mistakes are properties where the graph would correspond to a valid graph, but a molecule that corresponds to that graph cannot exist. So this would form a valid graph, but not a valid molecule. In that example, we would have oxygen, and before I mentioned oxygen can only have 2 bonds, but this string says the oxygen needs to have a double bond to carbon and a double bond to carbon here, so 4 bonds. But that is physically impossible, and therefore this string does not represent a valid molecule.
Mario, if I can interrupt you for a second, we had a question online. They were asking what you mean by property space.
Okay. Let me go back. So here by properties, I mean a function of the latent variable. So the latent space is a high-dimensional continuous space, and these properties are functions that map that space to a property that corresponds to the property of the molecule that corresponds to the point in the continuous space. So every point in the continuous latent space corresponds to a molecule, and this property space is the property of the molecule at that point. So it's Z is the latent space and F of Z is the property. For instance, a chemical property like solvability or how toxic the molecule is.
Sorry, Mario, is this is basically molecular property that you feed into the encoder, correct?
Yes. So for the training, we need to train. We need to train this neural network to approximate F of Z. So to approximate the property given a certain latent point.
Okay, thank you so much.
Okay, so there are several ways how one can construct invalid smiles, so smiles that do not correspond to valid strings, to valid molecules. Now, people have understood this problem quite quickly, and there are many ways how people try to solve it. For instance, one way is to change the way how the model generates molecules. So to change the model itself to forbid construction of invalid molecules. And this is a very active field. Sorry. Okay. And this is a very active field of research. Okay. And the problem of this approach is that those solutions are model dependent. Sorry. Okay. The problem is that this approach is model dependent. That means when you solve it for one model, for instance, you have a variational autoencoder that works well. If you come up with a different model, for instance, you want to use it in a GAN, you have to solve the same problem again for a GAN. So the problem is that's model dependent. A different way to solve the problem of invalid molecules is to change the molecular representation itself. And there was a very interesting work 2 years ago by O'Boyle and Dahlke, that introduced deep smiles. And deep smiles is a method to get around syntactical mistakes. So the syntactical mistakes that I was talking before, they cannot happen, or basically not happen anymore in deep smiles because of the way how it is constructed. The problem is still that these domain-dependent mistakes, the semantic mistakes, can happen. So semantic constraints are not satisfied here. And this is where our work comes in. With our representation, so we solved this problem by introducing a general representation for molecular strings, for molecular graphs. And we do it in a simple way by representing the molecules in a string-based way. But in our case, every string, every selfie string, we call this representation selfie, corresponds to a valid molecule. So every combination of letters in the selfie alphabet is a meaningful molecule. And one can think about this in a more general way. Namely, think about a language where every combination of letters corresponds to a meaningful word. And this is what SELFIES is for chemistry. And it is general enough that I explain it now in chemistry, but it can also work in quantum physics, in biology, everywhere where you have semantically constrained graphs. You can make a 100% valid representation of them. Okay, the idea of SELFIES is to use a formal grammar. A formal grammar is a concept from theoretical computer science, and it is closely related to automaton theory. So what a SELFIES string is basically an instruction set for a very simple formal automaton, or equivalently for a very simple formal grammar. So the idea is to encode this physically semantic constraints into the state of the derivation, such that during the derivation it's not possible to make any mistakes, because those states of the derivation carry a small amount of information that is enough to avoid all of those invalid molecules. And I would like to show you one simple example. Let's say we have this selfie string. We start in the state x, 0, and the first instruction is f. So f, we look up the replacement rule of this formal grammar for f is fx1. That means we replace x0 by fx1. Now we are in the state x1. So the next symbol that we derive is double bond C. We derive double bond C, and the replacement rule of the formal grammar is cx3. So now we are in the state x3. Again, double bond C goes to double bond C x2. We are in the state x2. We derive the next and last symbol, which is hash n. Hash n produces equal n. And we are finished with the derivation. And you see, as a chemist, you would see that this is a valid molecule. It satisfies all semantic constraints from the bonds. And if you look back to the selfie string, it appears here that that this would lead to a wrong molecule, because a fluor can only have one bond, but the carbon wants 2 bonds. But because of this additional information that's stored in this derivation state, the next symbol is only allowed to create 1 bond. And that is how the semantic constraints are inbuilt. The number of bonds is encoded in the state of the derivation. Now, this table looks quite complicated, but actually one can derive such a table in an automated way from a given data set. So this this table is 100% algorithmically defined. So given a large data set and specific rules to derive it, you can derive this whole table automatically. Okay, so we can now perform some experiments, which we did, and we took 4 different molecular representations. This is smiles, deep smiles that I mentioned before that solves syntactic mistakes. Grammar VAE is a VAE-based representation which can be also interpreted standalone, and our selfies representation. Now, if we use a valid large molecule and we perform a single modification of the string, the validity of most other representations drop quite significantly, while each of the output molecules in the selfies representation are valid. And a similar test can be done when you start from entirely random strings. Entirely random strings for smiles are valid in roughly 3% of the cases. And that is specific strings, I think up to 20 letters or so. They are valid in roughly 2 or 3%. Deep smiles also roughly 3%, while each of them is valid in the smiles representation. So every random string is valid in smiles.
Mario, if I can interrupt you for a second. The reason why you're emphasizing random validity, is it because of the nature of the problem that we're trying to solve, which is sequence to sequence problem?
Yeah. Yes. The task that we want to solve later is to generate new molecules with some generative models, and the output of those models should be valid. And if the representation is selfies without changing anything in the model, it's guaranteed to be 100% valid.
Okay, thank you.
Okay, so, and actually this is exactly what we tested in 2 cases. When we use those 4 representation in a variational autoencoder, just try to autoencode a large amount of molecules, we see that the validity of the output of smiles after the training is below 75%. For deep smiles, it's 81, and for selfies, it's 100% valid. So each molecule that we sample from the latent space is valid. If we look at reconstruction, also the reconstruction quality of selfies is much higher. And this is very important. The last part, the diversity of the latent space is significantly higher for selfies than, for instance, for smiles. That means inside of the variational autoencoder, there's an enormous pool of potential interesting molecules that are stored.
Mario, for evaluating the diversity, how do you quantify diversity?
We sample a number of molecules, I think 10,000. I'm not sure about this number, I need to look it up, but we sample a number of molecules and we translate it into smiles and then into canonical smiles, which are molecular strings that are unique. And then we compare what is the fraction of different molecules in this set. And we see for selfies, 82% of the set is different, while for other representation, a lot is the same, and that is because the model was able to learn mainly the smallest molecules, and therefore it outputs in most of the cases very simple, very small molecules.
Thank you.
And in a very similar way, one can use generative adversarial networks and perform the same task, trying to generate new molecules from given a data set. And if we compare smiles versus selfies without changing anything in the GAN itself, taking the simplest GAN that is possible, we see that our diversity is much, much higher than for smiles. Okay, now you asked the question about the diversity. I mentioned that the diversity is extremely important because it gives you kind of the potential that your machine learning model has access to. So how many molecules are encoded inside of the latent space is kind of your potential for finding a good molecule. And here on the x-axis is the region of space that we sample in the latent space. And here there's a logarithmic scale of diverse molecules. And for each point, we sample, we continue to sample until we cannot find new molecules in 10 samples in a row. So after we cannot find new molecules, we stop the sampling. And we see that for selfies, we have more than one, nearly 2 orders of magnitude more valid molecules, valid diverse molecules encoded in the latent space. So that is a huge potential for follow-up optimization in this space. And so these are now properties of encoding. We have also used the representation for actually optimizing some properties in a genetic algorithm that is enhanced with neural networks. And we are trying to solve the maximization of a certain property of a molecule. And what we did is using selfies as a representation. That means the genetic algorithm was just changing those selfie symbols, and we outperform most other approaches by far, and we got the highest value of a molecule for that specific property. And interestingly, random molecules, random selfie molecules, actually outperform several other deep generative model, which is quite surprising. But of course, the distribution of molecules is very different. Okay, so until now I was only talking about the first part, how to make a machine learning model that can solve a task better than a human. In this case, the task was finding good molecules. But now let me show one slide about how to get actually scientific ideas and inspiration from those models. How can we get how can we learn something about science from those models? And and for that, let me show you one paper that came out a few months ago from a group from ETH Zurich. So they used the variational autoencoder to encode position data, astronomical position data. Specifically, they encoded the information, the position information of the Sun and of Mars as they are seen from the Earth. And they want to reconstruct that values. And what they have used is disentanglement of variable. That means inside they change their loss function inside the latent space variable should be as disentangled as possible. And the surprising thing that they have seen is that while their input and output is in geocentric coordinates, the surprising thing is that the model internally uses heliocentric coordinates. Sorry. So internally, the model learns to use heliocentric coordinates. And that is totally remarkable because there was no force from outside or motivation from outside for the model to learn those different representation, this much more meaningful representation. Just this idea of disentanglement of variables. And now, in our setting, we also have variational autoencoder with a huge latent space, and and before the latent space, most of the latent space was invalid. That means didn't even correspond to some valid molecule. Here, some plots. These are blinds through the latent space where we sample molecules and we see the red parts are invalid molecules, green are valid. So for SMILES, most of the latent space was invalid. And there you don't even need to start interpreting interpreting what the model has learned when most of the latent space is invalid. But now for our new representation, and this is potentially the thing that I find most interesting about it, now 100% of the latent space is valid. And now we can finally try to understand what is the model learning inside about the molecules. If we apply techniques similar to this disentanglement of variables.
Mario, if I may ask a question, how do you how determine the validity of the molecules?
Good question. We use a very useful program called RDKit, and RDKit can create molecular graphs only if those SMILE string that you insert actually are valid SMILE string. So with RDKit, you can test whether a SMILE string is valid or not. And so we input SMILE string and we translate the selfies into SMILE string and test them with RDKit. And RDKit says each molecule that we sample for selfies is valid, but not for SMILES. Okay, so this already brings me to the end. With this new representation, for molecular graphs, or generally for semantically constrained graphs, which is simple. It's string-based. It is robust, which means every string of selfie is a valid graph. It is model independent. That means one doesn't need to change the variational autoencoder or a GAN or a genetic algorithm. You can just apply it directly. And also it is domain independent. Here I showed these examples for chemistry, but similarly, you can do the same thing also for quantum physics, potentially for biology. And the way how I think about it is that that it is a language where each combination of letters gives a meaningful word, which is quite surprising that this is possible. Okay, and one of the future steps that I'm really much looking forward is to analyze what the machine learning models learn. And that is now possible because our latent space is 100% valid. That means each point in the latent space corresponds to a molecule. And that should hopefully significantly enhance the potential for interpretability. Okay, and you can see the paper on arXiv. You can go to our GitHub repo, or you can directly install SELFIES via pip install SELFIES. Okay, thank you.
Thank you so much, Mario. So now we're going to see if there are any questions online. One of the questions asked is if you have an example or if you can explain how the grammar is acquired from the data.
Yes. So that is slightly more complicated. Just in short, we have the explanation of the algorithm in the supplementary. The idea is that you can see from the data you can see from the data directly the degree constraints. So for each vertex, you see how many edges are connected to this vertex. So the idea is first find all of your vertices. Now, when you have all of your vertices, for each of those vertices, go through the data set and count how many edges connect to this molecule, to this atom, or in general to this vertex. And if you have done this, you can easily derive automatically a rule set, a set of rules that has some state of the derivation that allow only for a given number of bonds. And if the SELFIES requests more bonds, you just don't allow them.
Okay, thank you. So we shortly discussed this, but for better understanding of the audience, your method, SELFIES, basically produces different strings based on different smile strings provided for the same molecule. I just wanted you to explain further as to why you made this decision. Because usually people discuss the fact that canonical smiles would make sense. So I'm just wondering why you made the decision of not producing canonical strings based on your method.
Mm-hmm. Okay, so the first thing is it was not a direct decision, but a consequence. The decision was to produce a representation that is 100% valid. Now, there's only a given amount of chemical space that is valid. That means if you have a representation that is similarly large as smiles, for instance, you can cover much more than that space. But we restrict ourselves with these derivation rules only to that space. And from that, it is already clear that with the same size, but this additional restriction, there will be cases that lead to the same molecule. For inverse design, it's not clear whether that's a huge problem. I've read this several times also, but it's not really clear for me why this is a problem. For instance, in the training, one can even do some tricks like increasing the training data artificially by using that symmetry, and from one smile string where I have the properties, I can produce several selfie string and train the network with all of those. Actually, a similar approach has been shown for smiles and was quite successful.
Okay, thank you so much. Let me just check if we have more questions. It seems like we don't have any more questions online. So with that, I thank you so much, Mario, for your time, and thank the audience for joining us today.
Thank you.